So there are 5 main mechanisms that bacteria use to evolve resistance to antibiotics. So the first mechanism, if your antibiotic is hanging around outside the cell, the first mechanism that bacteria use to evolve resistance is to actually just not take up that antibiotic. If the antibiotic can't get into the cell, it can't do any work. So you can decrease uptake. Another common mechanism that bacteria use is if the antibiotic is actually inside the cell, instead of decreasing uptake, what you can actually do is increase export. And we call this efflux. And a common mechanism of bacterial resistance to antibiotics is by developing what we call efflux pumps. And so these are little machines that live in the cell wall, and they pump the antibiotic from the inside of the cell to the outside. Another mechanism that bacteria use to avoid antibiotics is if the antibiotic's in the cell, instead of getting rid of it, you can break it down into different parts, and so it's now no longer effective. Basically, what we're doing is we're inactivating. There are several ways you can do that. You can modify the antibiotic, you can degrade it, you can chew it up, and in fact, some bacteria can actually break down antibiotics and then use them as a food source. So they're not only resistant to the antibiotic and to the treatment, it actually enables them to grow better and cause more disease. Another way that you can stop an antibiotic from working is if the antibiotic has to bind to a specific protein in the cell, you can modify that protein so that now the antibiotic will no longer bind. You can change essentially the target site of the antibiotic. So we call that target site modification. And then the final way that we can overcome antibiotics in the cell is instead of modifying the target, we can amplify the target. So that instead of only having one copy of the target in the cell, we now have many hundreds of identical copies. And so what happens here is that maybe the antibiotic can bind to 1 or 2 or 3 of them, and maybe when that binds, that inhibits the function of a couple of variants, but you still have enough copies of whatever the target is that the cell can grow, that that target can function. So different antibiotics have different preferences for these. So I mentioned already beta-lactams like penicillin. Beta-lactams, one of the ways that that microbes become resistant to beta-lactams is by decreasing their uptake. Aminoglycosides, similarly. One of the ways that bacteria become resistant to an antibiotic called trimethoprim, is by amplifying the target site so that it's now basically overwhelms the antibiotic and there's nothing that we can do about it. Pretty much everything can gain resistance by modifying the target site, including our friends aminoglycosides, including beta-lactams, including fluoroquinolones, including tetracyclines. Similarly, lots of bacteria can gain resistance by inactivating the antibiotic. And that includes, again, to the aminoglycosides, to the beta-lactams, to macrolides, quinolones, and to the tetracyclines. And then finally, increased efflux, getting the antibiotic out of the cell. Lots of things can gain resistance to different antibiotics by increasing efflux, and we can gain resistance to aminoglycosides, to our friends the beta-lactams, to our friends the tetracyclines, and so on. Okay? One of the things that's really critical to note when we talk about antibiotic resistance is that you can gain antibiotic resistance to penicillin by decreasing the uptake. You can gain resistance to penicillin by modifying the target site. You can gain resistance to penicillin by inactivating it. You can gain resistance to penicillin by increasing the export of the antibiotic. There's multiple ways that bacteria can become resistant to antibiotics. And there's multiple ways that bacteria can become resistant to the same antibiotic. And what makes it particularly challenging is that many of these mechanisms for resistance to antibiotics move around on plasmids, on phages, on mobile elements. They're shared between cells. So if one cell can gain resistance to an antibiotic, it will spread that resistance around, and all of the other cells will become resistant too.